Namaste viewers. Today in this video, you're going to understand about preparation of temporary squash from onion root tips. Before I begin, I request all the viewers to subscribe this channel to support. Thank you. This practical experiment during examination carry 8 marks. Okay, performance 5 marks, identification and diagram 3 marks. Now let us go through this today's experiment. Okay, in your syllabus, you are performing actually 10 experiments. In that second experiment today, we are understanding that is study of mitosis, preparation of temporary squash from onion root tips. I request all the students kindly make a note of all this information in your observation book. Before I begin today's experiment, let me give introduction related with the mitosis. All cells must replicate their DNA when dividing, right? During DNA replication, the two strands of the DNA double helix separate, right? And for each organ or for each original strand, a new complementary strand is produced, yielding two identical DNA molecules, right? DNA replication yields an identical pair of DNA molecules called sister chromatids attached at the region called the centromere. Dereplication eukaryotes is followed by the process called mitosis, which ensures that each daughter cells receive one copy of each of the replicated chromosomes. During the process of mitosis, the chromosomes pass through several stages known as prophase, metaphase, anaphase, and telophase. The actual division of the cytoplasm is called cytokinesis and occurs during telophase, right? During each of the preceding stages, particular events occur that contribute to the orderly distribution of the replicated chromosomes prior to cytokinesis. Kindly make a note in your observation book, study of mitosis, preparation of temporary squash from onion root tips. Put down today's date also. What is the aim of the experiment? Write down the aim: to prepare a temporary squash of onion root tip and to observe the different stages of mitosis. Materials required: onion root tips, forceps, needle, blade, cover slips, spirit lamp, tissue paper or blotting paper, microscope, compound microscope, 1% acetoarsine stain or lactoacetoarsine stain, in short LAO, 70% alcohol, 1 normal HCl for hydrolyze. What is the principle behind this experiment? Division of body or somatic body cells or somatic cells is known as mitosis. It is a complex process involving a series of changes leading to the formation of two equal daughter cells from a single parent cell. It was observed and named by Fleming. It is also called somatic cell division and occurs in all body cells. It is called equational division because the daughter cells have equal number of chromosomes. If I'm going very quick, you can pause this video and make a note in your observation book. Aim, materials required, and principle. If you're missing any kind of slide note, at the bottom you can see the serial number, okay, slide number. You can go to the slide number. Now let us go through. Note, take large sized mature onions about a week before the experiment. Cut all the dried roots from the skin, roots from the stem at the base of the bulb. Now place these onions on the mouth of coupling jar or any kind of glass jar filled with water in such a way that the stem portion must be dipped continuously in water. Okay, in about a week's time, new roots would develop and would start growing downward in the water. Take the onions and cut the milky white portion of root tips. Okay, is up to 5 mm length. We can preserve in cornet fluid or 70% alcohol, which dehydrates the roots. Okay, viewing mitosis in onion root tips. You can make good. This is the onion, right? Roots we have generated by dipping in the jar full of water, right? And here you can see the germination of the roots, right? These roots you are going to collect. When you take out this particular roots, here you can see this is the onion root tip, okay? Here, this is the protective root cap, okay? And above that, you can see region of cell division. Then we'll say this as region of cell elongation, okay? Why use onion root tips for viewing mitosis? The roots are easy to grow in large numbers. The cells at the tip of the roots are actively dividing, and thus many cells will be in stages of mitosis. The tips can be prepared in a way that allows them to be flattened on microscopes, slides, that is squashed, or you can also press by using the thumb or blunt-ended pencil so that the chromosomes of individual cells can be observed. The chromosomes can be stained to make them more easily observable. Regions of onion root tips. Okay, there are three cellular regions near the tip of an onion root tip. The first one here, the root cap. This root cap contains cells that cover and protect the underlying growth region as the root pushed through the through the soil. Second one is the region of cell division or meristem is where cells are actively dividing but not increasing significantly in size. In the region of cell elongation, cells are increasing in size but are not dividing. So we will not take this particular portion and we are very much interested with up to here. Okay, region of cell division is very, very important. That's why we're going to take, we have mentioned root tips. Tip region you are going to take out for the squash preparation of mitosis. Okay, note, onion, also known as the bulb onion or shallot, is referred to as Allium cepa. That is the botanical name. And you need to write this one in the italics or we need to underline the genus and species name. The chromosomes under study are taken from the meristem cells of the young growing onion roots. Okay. From that, again, onion roots, we are going to choose only the root tips. The objective of this experiment exercise are for you to better understand the process and stages of mitosis. Second objective, to prepare your own specimens of onion root in which you can visualize all the stages of mitosis. Okay. Now let us understand the procedure. Okay, how to perform this experiment. Okay, to perform this experiment, first you need to take the onion with newly sprouted roots and cut two root tips using scissors and transfer them to watch glass. Okay, to that watch glass, you need to add 1 drop of 1 normal HCl and 9 drops of lactoacetoarsine, or in short, acetoarsine stain. Okay, this 1 drop of HCl which is going to hydrolyze. The watch glass is then warmed over a flame till fumes arise. The root tips are then cooled, okay, taken on a clean glass slide with 2 to 3 drops of stain. Hope you are following. First, what we have done here, first we have taken the onion with newly sprouted roots, okay, and we have cut the 2 root tips using scissors and transferred them to watch glass. To that watch glass, we have added 1 drop of 1 normal HCl to hydrolyze and 9 drops of acetoarsine stain. Okay, and the watch glass is gently warmed over the flame. You need to warm the watch glass in such a way that directly you should not expose the flame. Just expose the flame and take out. Expose the flame and take out. Okay, till fumes arise. During heat condition, what happens? There perforation will happen through the transient perforation will happen through the transient perforation. This HCl will hydrolyze and 9 drops of this acetoarsine stain, the stain will enter into the cell. Once fumes arise, you need to stop exposing to the flame. The root tips are then cooled. Okay, just bring that one to the room temperature and transfer one root tips on a clean glass slide with 2 to 3 drops of stain. Okay, and when you transfer here on the glass slide, you need to take out only the root tips region. Okay, 1 mm tips region you are going to take, and rest of the roots you are going to discard. Okay, a cover slip is placed over it and gently tapped by placing it between the folds of a filter paper or blotting paper. Okay, we can gently tap by using the thumb, or you can also use blunt-ended pencil to tap. It is then observed under a microscope. After observation, draw pencil sketch of microscopic views or whatever you observe through microscope. First, observe on the 10x, then focus on 45x, and whatever you are going to observe the stages, make a pencil sketch. And in the examination hall, you need to show to the examiner. Any 2 stages, you need to call examiner and you need to show, okay, whatever stages you are going to get. Then later, whatever the stages you observed, comment on that particular mitotic stages. For example, I have got anaphase. Then write the circle. Okay, write the circle. In that circle, write anaphase stage. Okay, and then label that as anaphase. Okay, and comment on anaphase. Around 4 points you need to write for that particular anaphase stage. If you have observed metaphase, write the metaphase stage here, microscopic view. Then write, label that as metaphase and write 4 points related to metaphase, right? So you can comment on mitotic stages. Note, report the result to the batch in charge teacher or to the external examiner. Okay, after reporting, you must write the result. Result carries 1 mark. Compulsory, you must write. I have not showed that particular result 1 mark, but it is understood. You need to write the result. The mitotic squash observed and showed the prophase, metaphase, anaphase, and telophase stages. If you got only metaphase, anaphase, write only metaphase, anaphase, but try to look into the all the stages of mitosis. Okay, this is all about the procedure and the result. Now let us understand the practical view of this particular experiment of mitosis. First, I told in the procedure and even in the note, first you are going to take this kind of onion. Okay, remove the old dried roots. Okay, once you trimmed all this particular onion, then keep that in the jar containing water for the growing of onion root tips. So once you remove the old dried roots, this particular region, bottom region, that should be placed in a jar containing water. Okay, it means place onion in each tumbler or maybe coupling jar where only the lower surface of the bulb should be in contact with water. Okay, lower surface that should be contact with the water. After certain days, for example, after 3 days, you are going to get this kind of white roots. Okay, sprouted roots can be seen. This kind of roots you need to get, okay, for the scratch preparation of onion root tips. Then you are going to take only the tip region of this particular root. Rest will be discarded. Okay, to perform this experiment, you need acetocarmine stain or lactoacetoarsine stain, 1 normal HCl, then you need forceps, scissors, okay, then you need blade, spirit lamp, compound microscope, then slide, clean glass slide, and you need cover slips, then watch glass you need, okay, then you need blotting paper, then you need dropper, okay, and other requirements may required. And how to perform the experiment? First, you are going to take this sprouted onion root tips and you are going to cut this particular portion of the onion roots. And once you cut those onion roots freshly also you can use, as you can also preserve in the preserving agents. Okay, I have preserved here. This roots will be taken now. This roots will be taken and placed in the watch glass. Okay, I've taken from this particular jar, small container, and placed in this particular watch glass. After placing into the watch glass, you need to add here one drop of one normal HCl and 9 drops of LaO or acetoarsine stain we have added, right? After the addition, you need to expose to the flame, okay? Gently, you need to warm this particular watch glass to this particular flame. Once fume arise, you need to stop heating, okay? Then you need to take out by using the forceps, one root tips, okay? One or two or three root tips. You can transfer to the slide, clean glass slide with 1 or 2 drops of LaO stain. Okay, you need to transfer. After transferring, by using the blade, you need to cut only the tip region of the onion root tips, only the tip. For example, if this is the, for example, if this is the onion root tips, only this much region you are going to collect, only the tip region you are going to collect, and rest will be discarded. Okay, you are going to discard this one, only this much portion you are going to take. This will be taken out for the squash preparation. So this entire region will be discarded. Tip region will be taken here. Okay, as you can see here, I have cut here only the tip region I will be using. Okay, I can make out here. I have cut here the tip region and the rest region will be discarded. Okay, the rest region will be discarded. Okay, now we can see I have removed this particular extra additional root region. Okay, I've taken only the root tips. Okay, only root tips I have taken here. Then to the root tips, carefully by using the needle and forceps, okay, you can place the cover slip. Make sure there should not be any kind of air bubbles. Okay, I've placed the cover slip here, right? After placing the cover slip, what are you going to do? This particular slide you are going to place between the two folds of blotting paper, okay, to remove the excess stain and also to squash by using blunt-ended pencil or by using the thumb, right? That is, place a slide between the folds of two folds of blotting paper. Gently press cover slips with your thumb or blunt-ended pencil so that the tissue of the root tip spread as thin layer of cells. When there is a proper spread of thin layer of cells, then we'll be able to observe this proper spreading of the mitotic stages, right? Okay, after that, pressing by using the thumb or blunted pencil, observe under the microscope. First, observe under the 45 10x. After observed in the 10x, then focus under 45x. Okay, when you observe the 45, you'll be able to see this kind of particular view. Okay, this is the microscopic view. Okay, they need to observe like this. Here you can see, here you can see the telophase. Okay, and metaphase, anaphase, you will be able to observe. Then make a sketch, microscopic, below right, microscopic view of mitotic stages and make a pencil sketch of mitosis. Okay. Then in the microscopic view, you'll be able to observe interphase, that is cells in this stage, that is resting stage, are easily identified by their prominent nucleoli. Okay. Other one is prophase. This is early prophase. Here you can see the chromosomal condensation initiates, giving the nucleus a grainy appearance. Okay, that is nucleolus is still visible. Then metaphase, you can see very beautiful metaphase. You are going to observe like this only in the microscope. Okay, chromosomes align along metaphase or equatorial plate. This is metaphase. Anaphase, this is early anaphase. Okay, it's still starting. Chromosomes with separate separated sister chromatids begin to move away from the equatorial plate towards opposite poles. In equal numbers, they will separate and move into the different opposite poles. Okay, you can see very clear anaphase. Sister chromatids reach opposite poles in equal numbers. Next one is telophase. You can see this is the early telophase. Okay, and this is the late telophase. Telophase, that is an early telophase, chromosomal decondensation initiates. Here in this case, chromosomes almost fully decondense to form original chromosomal network. And here you can see the late telophase, that is early cytokinesis. Growing cell plate can be observed, which is going to separate the two cells. Chromosome individuality is lost, forming chromatin network, nuclear membrane, right? And nucleus reappears and cell plate begins to form. Okay, now let us go through the discussion on mitotic stages. Whatever you observed in previous slides, these stages. Now let us discuss about all those stages. Let us go through the first one, interphase. Interphase is a series of changes that takes place in a newly formed cell and its nucleus before it becomes capable of division again. Okay, it is also called preparatory phase or intermitosis. Okay, typically interphase lasts for about at least 91% of the total time required for the cell cycle. Okay, in cell cycle you are going to understand in detail about different stages of cell cycle. Interphase proceeds in three stages, that is G1, S phase, and G2 phase, followed by the cycles of mitosis and cytokinesis. Okay, the cell's nuclear DNA contents are duplicated during S phase. Okay, during S phase, you can see this particular mitosis formation. Interphase, this is the period between two mitotic divisions where both nucleus and cytoplasm attain maximum growth. Okay, the following process occur during interphase. What are those processes? Synthesis of DNA and protein can be observed. Then DNA replication leading to chromosome duplication, building up ATP, which is used for different stages of mitosis. Okay. Now let us go through some important features that can be identified during this stage are nucleus and nucleolus appear conspicuously large. Then nuclear membrane is intact. The duplicate chromosome are visible as extremely thin strands. Mitosis consists of the following four stages after the interphase, that is prophase, metaphase, anaphase, and telophase. Now let us understand here the first stage after interphase, that is prophase. Okay, here you can see the prophase. The chromatin material condenses into distinct chromatin threads by dehydration. Okay, the chromatin threads gradually become short and thick due to specialization and dehydration. Each chromosome is formed of two sister chromatids which are held together along their length by a centromere. The nucleolus and nuclear membrane disappear by the end of prophase, right? After prophase, we have metaphase, right? During that time, you won't be able to observe this nuclear membrane and nucleolus. They'll disappear. The centrioles duplicate and move apart to occupy the opposite poles of the cell. Then microtubules appears across cell to form spindle fibers. In short, if you want to write information about prophase, you can write this much information enough. During prophase, the chromosomes supercoil and the fibers of the spindle apparatus begin to form between centromeres located at the pole of the cells, right? The nuclear membrane also disintegrates at this time, right? During prophase, nuclear membrane will disappear, freeing the chromosomes into surrounding cytoplasm, and that will be ready for the next stage, that is metaphase. Here you can see all the chromosomes which are aligned in the center, where centromeres are in the equatorial region or in the center region, right? Metaphase is identified by fully formed fibers. Due to heavy coiling and dehydration, the chromosomes become short and thick, and two chromatids separate except at the centromere. Okay, all chromosomes move towards the center of the cell and orient themselves on the equator of the spindle. In such case, the centromere lies in the equator, right? And asters are oriented towards the poles. The centromere of one chromatin faces one pole and the other faces the other pole, and they move slightly apart, and they're getting ready for the anaphase stage. Okay, during prometaphase, some of the fibers attached to the centromere of each pair of sister chromatids, and they begin to move towards the center of the cell, right? At metaphase, the chromosomes have come to rest along the center of plane of the cell. This is all about the metaphase. After the metaphase, the cells are ready for the anaphase, right? You can see all the chromosomes which are in the opposite pole, right? You can see the opposite pole. The centromere of each chromosomes divide and allow the separation of two sister chromatids into two daughter chromosomes. Okay, the daughter chromosomes move apart and migrate towards opposite poles. You can see here the daughter chromosomes which are moved apart, and one is in the other pole, other is in the other pole, right? The movement of chromosomes is governed by the contraction of spindle fibers. Equal number of chromosomes are present which are placed at opposite poles. Moving daughter chromosomes exhibit different shapes depending on the position of the chromosomes. During anaphase, the centromere split and sister chromatids begin to migrate towards the opposite poles of the cell. Once anaphase completes, this particular cell is now ready for the next stage, that is telophase. Here you can see this is the early telophase and this is the late telophase. Here, the chromosomes reach the opposite poles and form two groups. The chromosomes gradually undergo rehydration to form chromatin. Nuclear membrane, New nuclear membrane is formed around each group of chromosomes, right? Nucleolus is reformed and spindle fibers break down, right? All the spindle fibers which are break down and are absorbed into the cytoplasm. Okay, you can also write this summarized information. During telophase, the chromosomes at either end of the cell cluster begin to cluster together, which facilitates the formation of new nuclear membrane. This is one cytokinesis occur, right? The cytoplasm will divide, leading to two separate cells. One way to identify that telophase has begun is by looking for the formation of the cell plate. The new cell walls forming between the two cells to become two daughter cells. Here you can see the overall view of the mitotic stages, right? This is the interphase, and this is prophase, metaphase, anaphase, and telophase. Okay, this is how you are going to observe in the onion root tips microscopic view. Okay, here you can see cytokinesis differentiation between the plant cell and the animal cell. This is the plant cell and this one is the animal cell, where you can see the cleavage furrow can be seen in the animal cell. Whereas in the plant cell, you have the cell walls. You can see the formation of the cell plate. Okay, cytokinesis is the final stage of eukaryotic cell division, right? It occurs differently in animal, which is left process, and the plant, which is the right process in plant cells. During cytokinesis, the cytoplasm splits into two and the cell divides. In animal cells, the plasma membrane of the parent cell pinches inward along the cell's equator until two daughter cells form. Okay, that is through the cleavage furrow. Whereas in the plant cell, a cell plate forms along the equator of the parent cell. Okay, then a new plasma membrane and cell wall form along each side of the cell plate. Okay, you know the difference between animal cell and plant cell. These animal cells, they are devoid of cell wall, and plant cells, you can see the presence of cell wall. So here you can see the formation of the cell plate. This is how you are going to observe under the microscope when you use the lactoacetoarsine or acetoarsine stain. This is the interphase, right? This is the prophase. Then you can see this is the metaphase, anaphase. Okay, this is the early telophase and this is the late telophase. Okay, this is also another view, the microscope. Here you can see the metaphase and here this is anaphase. Okay, here you can see again the telophase. Okay, here this is the anaphase. Here you can see the interphase. This is prophase. This is metaphase. And you have one more metaphase. Here one more metaphase. And here anaphase you have. Here one more metaphase. Okay, I'm going very quick, means you can pause this video and you can observe this particular slide. This is the overall view of mitotic stages. Interphase, early prophase, this is prophase, late prophase, this is metaphase, anaphase, telophase, formation of the cell plate here, then cytokinesis after division of the cytoplasm. This is all about the mitotic stages. At the end, you need to write the result. The mitotic squash observed and showed the prophase, metaphase, anaphase, and telophase stages. Hope you understood today's concept, preparation of mitotic squash preparation by using the onion root tips. I, Dr. Pratibha K, signing off for today. Stay tuned with me. I'll be back with some more videos. And thank you for watching.